Welcome, everybody. Good morning or good afternoon, depending on where you are in the world. I'm very happy to welcome you to a webinar today about TRxA's Bridge Awards. And of course, we like acronyms, so let me start with explaining what this all stands for. So we're the Critical Path Institute, or CPAth, Translational Therapeutics Accelerator, and we are here to present a bridging research and an innovation in drug development grants. So we're really happy that you all have joined in. Sorry, I have a frog in my throat, apparently. Good timing. But let's start with some introductions of the TRxA team. And Mark, let's start with you.
Yeah, so my name is Mark Drew. I'm the Director of Drug Discovery and Development. I'm a medicinal chemist. I spent most of my career, about 20 years at Sanofi.
Thank you, Mark. We're very glad to have you as part of the team. And then Michelle.
Hello, good morning, good afternoon, everyone. My name is Michelle Morgan. I am Associate Director of TRxA, overseeing the operational aspect of our program.
Wonderful. And then Kyla.
Hello, everyone. Good morning, good afternoon. My name is Kyla Edding. I'm the project coordinator for TRxA, and I help Michelle handle the operational side of things.
Thank you. And Kyla is the technical whiz behind the webinar today. So thank you, Kyla, for keeping us all organized and virtually connected. And my name is Maaike Everts. I'm the executive director of this program. Spent most of my career in academia at the University of Alabama at Birmingham, where I also led a drug discovery program. And it's been really a pleasure to be part of the Critical Path Institute and leading the TRxA program. So we'll jump right in and talk to you about what our mission is. So we are a global drug discovery and development program, and we support academic scientists in advancing new cutting-edge therapeutics from the lab to the clinic. And there's a couple of bolded words here in our mission that I wanted to briefly elaborate on. The first is global, and as you're from all parts of the world joining in with us for this webinar, we can fund projects all over the world. So that's really a nice feature of our program, that no matter where you are located, we're happy to help you. We're supporting academic scientists. So for us, it's important that you are still within either university or a nonprofit research institute, or maybe a research hospital. We cannot fund startup companies, so I want to make that clear on the front end. But if you're still within an academic institution or equivalent, we're happy to help you as well. And then we are focusing on therapeutics. So we're not supporting diagnostics or biomarkers or medical devices. So therapeutics or drugs is what we're currently focusing on. And within that, we have a more narrow focus. So currently, and for this RFP that we're talking about today for the bridge grants, we are supporting small molecules that are at a stage of lead optimization or IND enabling studies. We'd like to see composition of matter IP either in place or obtainable. And we also would like to make sure that the IP is still within your institution, so it had not been out licensed yet. And we'll talk about some of these aspects in more detail in the coming hour. We're welcoming any questions that you may have. Feel free to put them in the Q&A. We'll answer them at the end of the webinar. Michelle will actually go through them and be our Q&A wizard. So looking forward to hearing any questions that you might have. We also, towards the end of this talk, will go through some frequently asked questions that will hopefully already answer some of the questions that you may have. So let's talk about the valley of death. I think everybody that's tuning into this webinar is very familiar with this valley of death. You may be in the middle of it. And that's really referring to that space between where academic research kind of stops and industry likes to take up a project to further develop into clinical trials in patients. There's a lot of reasons for this valley of death. There's a really nice paper, the reference is on the bottom right of the slide, that goes into more detail about some of those aspects. And some are enumerated at the bottom of the screen. For example, reproducibility is an issue. We'll talk about that in a little bit. The technical expertise of people that work in drug discovery, many times still in academia, we tend to be in our own little fields, and it's really a team sport, drug discovery and development. So you may need expertise from somebody that's not in your immediate field, and how do you find them? There's intellectual property issues, there's funding issues. Some of this is not hypothesis-driven research. So how do you find a funding body that wants to support this kind of work for you? There's high risk. There's, it just takes time. So a lot of these things kind of conspire all together to result in this valley of death. There's also a lack of knowledge about regulatory science. There's things like, let's set up a target product profile, or TPP. This is a really useful exercise to do, even when you're preclinical, to think, okay, what's my target population going to be like? What are some of the clinical parameters that we're going to use at readouts for our clinical trial results? What kind of assessments do we need to do? And those kind of target product profiles, how will it be administered? Is it an oral pill? Is it an injection? Those may actually inform some of the decisions you need to make earlier on in the development pathway. It's good to start thinking about these things early on. Then there's questions of how do you even submit an IND? What parts are needed? When I was in academia, I was involved in some of these IND applications, so I had to learn a lot about CMC. Like, what does CMC even stand for? It's chemistry, manufacturing, and control, by the way. But still, there's an expertise there, and there's a layer of other, areas of knowledge that you really need to tap into to really see that whole picture of where you currently are to getting something to the clinic. Appropriate clinical endpoints, what's accepted by the agency. And then there's also a lot of support programs actually out there that you may not be aware of. For example, at the FDA, there's an Oncology Center of Excellence Project Catalyst, and they facilitate what's called Accelerator Innovator Discussion Meetings. There's also a learning platform there called the Oncology Regulatory Expertise and Early Guidance. A lot of this is typically not really on the awareness of an academic investigator. So it's really a lack there that, something that could help you, but if you don't know about it, then, well, you can't tap into it. There's also an example at the European Medicine Agency. They actually give free scientific advice. So normally with the EMA, scientific advice is not free. But if you work in orphan and pediatric indication, actually that fee is waived. A lot of us in academic nonprofit research areas work on those orphan and pediatric indications. So again, knowledge about these kind of regulatory aspects can really help accelerate your program. So let's talk briefly about reproducibility. This is an issue as well, and it is brought up a lot by, for example, venture capitalists or industry or pharma partners that are looking at an asset that they may want to license from you. And it's a risky thing because actually quite a lot of academic studies cannot be reproduced. Now, that's not to say that we're all being nefarious and duplicitous, but there's actually a really nice paper here, and I have a screenshot on the left. It's called "Why Most Published Research Findings Are False," which I'm sure was being, phrased a little provocatively to get lots of readers. But actually, it's a really good paper, and they talk about some statistical consideration, actually, why some research findings may be less likely to be true. There's a small sample size, if there's a small effect size, if there's a lot of flexibility in how you design an experiment, how you define outcomes, how do you analyze them, if there's a lot of financial and other interests, and if the scientific field is hot. And you can imagine that a lot of these aspects are actually relevant for academic science. So there's a really nice cartoon here that I found on Nature, where you look at even a robustness of an assay of if you do something in June versus later in the year, or different technicians are executing the program, there's a different batch of mice, you may actually even get different results. So all of that plays into that perceived, it's risky to trust into a one lab doing one kind of experiment before you would decide to maybe invest in a program. So accelerators like TRxA can help with that. So it's all about reducing risk. And this is accelerators in general, and we'll talk a little bit more, actually a lot more about TRxA in particular. But in general, there's different aspects of these accelerators that can, for example, help with the culture of collaboration, put together interdisciplinary project teams. What we see a lot is when projects that come to us that have already pre-existing collaborations between, let's say, a biologist and a chemist, and maybe a clinician, having all those different aspects and different eyes on a project tends to elevate the quality of the project. So accelerators like ours try to put together these interdisciplinary project teams so that we can really get the best quality of the advice for a project coming from all kinds of different expertise. Then it's the aspect of what target do you go after? How do you select which one is a good one? So druggability plays a role. Have has this class of targets been drugged with the molecule before? What's the role of the target in the disease? So accelerators, where you have to apply to, get funding, it really helps you, hopefully, to define that role. And then, you can see with the scientific advisory board, it looks at a spectrum of these targets. The ones that tends to have the least risk tend to emerge to the top. We can also help with how do you design your assays, and what are some orthogonal assays, meaning you're looking at the same kind of endpoint, like is my drug working or not, but with a different type of analytical method. Because sometimes there may be artifacts in certain types of assays that, accelerators like ours that see a lot of different programs are aware of, but you as an individual may not actually be so familiar with. So again, having multiple eyes on the project, multiple types of expertise can really help there. There's the opportunity to get medicinal chemistry support, and we're very lucky that Mark is part of our program with this chemistry background, spent a lot of time in pharma and biotech. Again, it's having done this before, seen it before, help to define how do we actually characterize these compounds, which compounds need to have what kind of analysis done on them, because you probably can't do everything on every single compound that you make, right? So we can together be part of a team in which we have those kind of discussions. Then there's the definition of, okay, can we engage a target? Can we look in, for example, an in vivo model that a drug is actually having a pharmacological effect? What kind of selectivity evaluations do we need to do? And that's all depending on what's the target, what's the molecule like, what is the disease like, all kinds of aspects. Then we also like to encourage early engagements with pharma and biotech to get feedback. So if you talk with your project and you go to speak with 3 or 4 different potential interested licensees, and they all give you the same question, like, hey, have you looked at this, right? Have you done this experiment or that experiment? Then you know there's something that you need to address. And you also get a sense of the interest of the market and the financial commitment that companies might be able to bring to the table. So having those conversations, and that's usually in conjunction with your technology transfer office, is really helpful. And then accelerators like ours, but also others, could have some requirements in place to get some regulatory science input. For example, we are now asking that everybody that's funded by us puts together a target product profile. There's a document I was talking about a little bit earlier that kind of tells about, okay, what's the target population? How often per day do you need to take it? What's the desired outcome? What's the acceptable side effect profile? So again, having those brainstorming discussions around what actually needs to be in a TPP sometimes helps to clarify where you need to start, even if you're on a preclinical stage. So let's talk a little bit more in detail then about TRxA, or our Translational Therapeutics Accelerator. So let's start on the left. We talked about the problem of the transition between academia and industry is the drug development valley of death. Multifaceted, but lack of understanding of what kind of data package is needed to be of interest to industry partners is also a part of that. And then we spoke already a little bit about the regulatory aspects of drug development. It's not something that you're typically exposed to in academia. So why would you know that, right? But knowing what you don't know is already very helpful. And then you have the issue of academic studies sometimes lacking reproducibility. So then looking at the middle column here, what we would like to provide is both funding, but also expertise. So we don't just write a check, but we're really going to be partners in the program. And we can provide some regulatory guidance, but also assist with study design and implementation. We're doing that with some of our current projects where there may be a couple of different avenues that we could take. Which one is the most strategically prudent? And having those conversations, again, with the multidisciplinary approach is really helpful. And then we support involvement of contract research organizations, or CROs. So it's kind of twofold. One is to validate the academic findings. So if you have a key experiment that's really wowing everybody, that ultimately would hopefully further invest in your project. If you have a CRO or another neutral third party execute that project and replicate your findings, this will do a lot for the confidence that potential future investors have in your project. So we definitely help support replicating of those key findings. The other part is that a lot of CROs can do some parts of the work faster, probably cheaper and better than something that could be done in an academic lab. One of the examples is, for example, pharmacokinetic studies in animals. This can be done relatively cheaply at CROs rather than doing it in-house with having to define an analytical method, making sure that if you do intravenous administration, you actually get into the blood vessel, right? If you have students having to learn it, there's a lot of factors that can complicate those kind of experiments within the academic realm. Whereas if you use a CRO, you probably get the results within 2 weeks. So then let's go to the right side of the slide. A couple of aspects about how we are unique. So we're part of CPATH, or the Critical Path Institute. So we're an independent nonprofit entity. We do not take any ownership or claims on IP. That all stays with you or your institution. And then we'll talk a little bit more about the critical path and all our regulatory knowledge that we have and relationships, so we can leverage those relationships and get that knowledge to you, to the project. And then we can help provide some project management and project planning. What tends to help is that if we have regular meetings, we meet monthly with the people that we fund, and it just keeps things on track. When there's a roadblock, we can together talk about it, how to overcome this obstacle. And it's been proven to be really helpful to have that frequent check-in. And they're not punitive check-ins, they're really actually fun meetings. You talk about science, you talk about next steps, and we can really help streamline that process to always keep that end goal in mind, which is getting to a patient, getting to IND. So then when you look at the pipeline at the bottom, at the graphic, where TRxA plays a role is in the lead optimization IND enabling studies phase of the drug discovery and development pathway. So that means we do not fund drug discovery as in, for example, high throughput screening. We don't really fund target identification. But if you come to us with like, hey, I have a good target, and this is the biological and scientific rationale why this target plays a role in disease, and I have chemical matter, and having some analogs maybe around your lead compound already, that's really the sweet spot where we would like to engage. And then we can help you throughout IND enabling studies, but then once it reaches a patient, that is when we step back again and somebody else will hopefully take over. We'll talk a little bit more about the entry and exit criteria of our different funding types that we have. But that gives you kind of a big picture. It's kind of the chemistry IND enabling stage where we'd like to play a role. So we are funded ourselves by a nonprofit foundation called the Frederick Gardner Cottrell Foundation. And this was established by Research Corporation Technologies, or RCTech. And this is a venture capital group in Tucson, Arizona. So they established a foundation a long time ago in the late 90s, and they provide financial support for a scientific and educational program at nonprofits. So they gave their largest grant that they've given out in their history to the Critical Path Institute to establish our accelerator. So then let's talk a little bit about the broader critical path context. So let's look at the graph on the right. So we play in a pre-competitive neutral ground where we bring different stakeholders together to collaborate on aspects of drug development that you cannot really do on your own. So for example, we have academic collaborators, we have a lot of patient groups actually come together, and they're in consortia that tend to focus on a particular disease. Not all, but many of them. We work with, for example, the CDC, WHO, industry members, but then we also work very closely with regulators. So the FDA are a big part of it, but also the EMA, as well as some other regulatory bodies around the world. And all of those consortia, again, they bring these different stakeholders together to focus on drug development tools, or DDTs, in their regulatory endorsement. So that an example could be, can we define a biomarker that predicts kidney injury in the preclinical space before it even reaches a human? Or can we use wearable devices like your smartphones or your watches for Parkinson's patients to get more data from a Parkinson's patient in their daily lives that can then be used for a clinician to make treatment decisions based on that? Can we come up with clinical outcome assessments that are either patient reported or maybe clinician reported? It can be used as an endpoint in a clinical trial. It's those kind of broad questions that all these stakeholders come together and a consortia facilitate getting answers to them. And then because the regulators are at the table, you immediately have that feedback whether such a tool is being developed will be acceptable by the regulators when you submit either an IND or an NDA. Now, TRxA is a little bit different from many of our colleagues' programs because we directly support financially and with knowledge the development of drugs. So we can tap into our colleagues and our consortia's expertise to get subject matter experts that may help provide an extra set of eyes. For example, if we really need a toxicologist to provide some input on a project, we will have a toxicologist within our network that we can pull into the project and provide some advice. Or if it's a disease-specific expertise that you need, whether it's type 1 diabetes or kidney transplant. We have 26 or so consortia now, I think, that really cover a wide breadth of diseases and indications in therapeutic areas. So TRxA then can apply CPAT's technical as well as regulatory expertise for our funded projects. And then we also, again, help with that project planning and project management to move projects towards the clinic. So let's talk about the types of awards that we have. Oops, I went too far. So we have three called Stage 1, Stage 2, and Stage 3 bridge awards. Let's talk about the first one, Stage 1 initially. So this is for $250,000 US. Now, again, we can fund anywhere in the world, so we would convert that to your local currency. This is total cost, and we do cap indirect costs at 10%, as many other nonprofits do as well. But this Stage 1 type of bridge award is for early lead compound to late lead series development. So again, you may come in with, hey, I have a molecule, I've made a couple of analogs around it, so I can see that I can optimize it, but I really need to work to get towards a true late lead series. That's Stage 1. Stage 2 is when you have that late lead series already, you can come in that stage to us as well, but you really need to hone in on the clinical candidate. There's probably something you need to fix about your molecule, whether that is solubility, whether it's oral bioavailability, selectivity, you name it. There's probably something that's not quite right before you wanted to go into IND enabling studies. This will be funded for, again, the total cost of $500,000. This is all for 1 year. And in Stage 3, this is when you already have a candidate, you've selected it, you know this is the one you want to take into humans, but you need to do IND enabling studies. Now, that could be API scale up, or it could be a GLP tox study in dogs. Right? There's a lot of very expensive mandatory aspects to IND enabling studies that we would like to help support. And that will be for $1 million. Again, total cost for 1 year. Now we know that 250,000, 500,000, or 1 million is not enough to get something from the bench to the clinic. It takes more money than that, right? We're not naive. But we love to collaborate with others, with other funders. And also we'd like to think that these amounts of money do move the needle enough to, again, create this data package. Is that going to be interesting to the funders that come after us that may have deeper pockets, whether that's a venture capital group that wants to invest in a startup with this asset, or whether that's an established biotech or pharma company that wants to license it from the university. So let's go into a little bit more detail about these different stages. So the Stage 1 Bridge Award. Now, and you should read these kind of as job descriptions. So, we all know you don't have to hit every point in a job description 100% before you apply for a job, right? It kind of applies here too. So it gives you a sense of what we're looking for. But if you miss one bullet or like, oh, kind of not quite there yet, I would say engage with us anyway, because if everything else looks really strong, then we certainly would take a look at it. At Stage 1, the entry criteria are that the project, again, is in early lead optimization. You need to have a tractable drug lead, and ideally more than one, so drug leads from multiple chemical series. Now, the reason we would like to have multiple chemical series, so more than one, is because if there's a fatal flaw with your initial compound, your initial series, and you really hit a roadblock, then having a backup series is really helpful to be able to continue the project. Now, we would also like to see that you've done some structure-activity relationship optimization, or at least knowing that you can optimize something. That is not like if you change even one thing anywhere in a molecule that now it doesn't work anymore. So having a few analogs around this. Now, this can be analog by catalog where you've bought a couple. They're not perfect analogs, but at least it shows that it has some activity, maybe some selectivity that you can already look at. So having a little bit of information and knowledge about your molecule, your scaffold, and how much you can play with it. You need to have in vitro pharmacology assays in place, whether it's biochemical or cell-based, looking for potency and selectivity. That's really critical because we need to be able to evaluate those molecules. And then you need to have access to an available or conceived in vivo pharmacodynamic model. Now, what we mean here is that you don't need to have in vivo data before you come to us. You can have a plan to get it, but you need to tell us what that plan is. What model are you going to use? Is there a CRO that can do that? Do you have a collaborator that has that? Do you have it in your lab? And we're looking for a pharmacodynamic model, as in we want to show target engagement. We want to show that the compounds that we're developing have a pharmacological effect. Now, this does not necessarily need to be your efficacy model. An example is Parkinson's disease. All the animal models there, a lot of them have different flaws, different opinions about which is the best one. But an in vivo pharmacodynamic model could be, hey, if we're trying to inhibit a certain enzyme in a pathway in the brain, let's check whether if we inject a drug into a mouse, we get the brain out and we see, did the drug inhibit the enzyme, yes or not? That would be appropriate at this stage in the game because it shows target engagement. But then later in the game, once you're in stage 2 or stage 3, that's where you would do your efficacy experiment. And more complicated models that tend to take longer as well. So then success criteria of these for stage 1 bridge awards is that together with us, you would have defined a target product profile. There would be optimized lead molecules from multiple series with characterized in vitro pharmacology, including cell-based activity, characterized ADME properties. So it can be done mostly in vitro, like, microsome stability, for example, but also maybe some initial rodent, whether it's rat or mouse, PK in vivo. And then we would like to see demonstrated in vivo pharmacology, again, in this pharmacodynamic model. Now, at this stage in the game, you need to really have access to or conceive of your efficacy model, though, your in vivo efficacy model, because that comes into play in the stage 2 awards. So stage 2, the entry criteria are actually the exit criteria for stage 1. And this is something that a lot of people ask us as well. Well, if I come in stage 1, will I be eligible to then get a stage 2 and stage 3? And the answer is yes. So you don't need to worry about pigeonholing yourself too much. What actually will happen is once you've completed one funding cycle with us, we'll do this on an ad hoc, more bespoke method rather than you competing again with everybody else that's applying for stage 2 or stage 3 award. So again, entry criteria for stage 2 are defined target product profile. You need to have optimized leads from multiple series with in vitro pharmacology, some ADME properties characterized, and an in vivo pharmacodynamic model readouts, and then having an efficacy model in your back pocket. So then the success criteria after you've done a stage 2 bridge award for a year is that at this point in time, you probably will have defined your target product profile, but also have a vetted regulatory plan to achieve the target product profile, and then optimized molecules that really meet candidate selection criteria. So again, characterized in vitro and in vivo pharmacology, demonstrated efficacy in vivo, characterized ADME properties in vitro, and again, rodent, and maybe at this point in time, non-rodent in vivo data, characterized toxicology properties, and then a defined non-clinical formulation. What are you going to use for your IND-enabling studies? At this point in the game, you also should be thinking about what's your API, your active pharmaceutical ingredient, scale-up and characterization plan. As chemists will know, making 10 milligrams on the bench is very different than making a kilo in a big reactor. So sometimes you need to get different synthetic routes. So all of that process chemistry really is something that needs to get attention at this game, name it game. So let's go to stage 3. So again, the entry criteria here are the same success criteria for stage 2. The defined target product profile and a vetted regulatory plan of that, optimized molecule meeting these candidate selection criteria, and then having that defined GMP API scale up and characterization plan. And at the end of that year, we would like to see, of course, that the target product profile is being adhered to and the regulatory plan really have a well-characterized molecule at this point in time with a complete toxicology package that would enable first-in-human studies, so general safety and genetic toxicology, and also have probably an optimized GMP API scale-up strategy. So we have a 3-step annual selection process. The first one is the one that's active right now, which is a call for pre-proposals. So this opened last week, January 22nd, and the deadline is March 31st. We have established a Salesforce portal. So on our website, you can get a link to that portal and create an account. And then there will be a form on that portal that we ask you to fill out. And it's really a high-level description of the project that we're looking for at this point in time. It should be all non-confidential summary information. So, for example, what's the molecular target? What's the properties of your lead molecule? Really big picture items. These answers are reviewed by an internal group here within the Critical Path Institute by our Programmatic Review Board, or PRB. So this PRB determines which projects then advance in the process to the next stage. So the next stage is that we request confidentiality agreements. So we go under CDA with your institution, and that way we can then ask you to upload and share confidential information with us. For example, the structure of your molecule. And Mark, he was, quite short in his own description, but he's also a patent agent. So together with his chemistry expertise and some quick searches in the patent literature, we just want to make sure that your chemical molecule is a good starting point for both IP and chemistry reasons. And then when you go through this process, and most people will pass this without too much trouble, you get invited for a full proposal. And this invitation will come out the week of July 1st and will be due August 26th. So it's about 6-7 weeks or so that you have time to put this full proposal together. Now, throughout all of this, actually not just in this full proposal stage, but also earlier, we as the TRxA team are available for you to provide coaching, mentoring, input, answer questions that you may have to help put together a plan. So we're not reviewers on purpose. We outsource that to other people because we really want to help you put together the best pre-proposal or full proposal that's possible. Now, of course, it depends on bandwidth and how busy we are at that moment, how much information and advice we can give, but we really like to try to help as much as we can. So these full proposals are reviewed by an outside scientific advisory committee. We actually have members of these committees listed on our website, so we're transparent about that. And they will score the proposals. And they are a group of people, both in the US and Europe, that have a wide variety of expertise. We have a toxicologist on there, clinician, to a venture capitalist, retired pharma executives, different therapeutic areas that they have knowledge about. So they review all the proposals, but then also for each proposal, we try to get an ad hoc reviewer from a patient organization that has scientific expertise, because we really want to hear that patient voice as well when we review proposals to see which ones should we select for funding. So the scientific scores get aggregated, they get forwarded to our PRB, our Programmatic Review Board again, and they make, based on scientific merit, of course, they make decisions on who to fund, but sometimes there's more scientifically meritorious projects than we have ability to fund, so then there's some strategic fit decisions that need to be made as well. So the PRB does that, and that will happen in the late fall. So we should be able to announce who gets selected for funding around October, and then we initiate contract agreement with your institution. So here's a couple of FAQs. The first one is about who we fund. So who's eligible for TRxA funding? So you need to be faculty or equivalent at universities or a nonprofit institution, but you can be anywhere around the world. So unfortunately, we do not support postdocs or other trainees. And just because you're probably going to move, if you're a postdoc, you're probably going to move to another institution. So we like to fund faculty where there's a little bit more stability there. Are there any institutional requirements? So as long as your project is an academic or a nonprofit organization, you are good to go. Now, sometimes there may be some gray areas where somebody says, like, well, I'm in this hospital. And so if you're a little bit confused, like, do we qualify or not, just reach out to us and we will help you make that decision. And then there's a question, well, are there any institutional commitments required if I get funded? So we do expect the institution to engage with us and negotiate a reasonable project agreement. And we have actually a template of this on our website. So actually we encourage everybody that work with your grants and contracts office or your tech transfer office, go ahead and download that template so that they can take a look and see if the terms are in principle agreeable to them. We've put several of these in place already with several types of institutions, and it hasn't been an issue, but we like to be as transparent about it up front as possible. So what do we fund then? So do you have criteria for what is a small molecule? And we get this quite a bit. Are peptidomimetics allowed to have a peptide? How about that one? Peptidomimetic would in principle qualify for funding, but it still should be small-ish. So we like to ask that the molecular weight is around 1,000 Daltons or less. And of course, there's some gray zones there. But we like to shy away from large peptides. But there's an example I worked with when I was actually in my previous institution where we started with the peptide with 4 amino acids, and this was transformed into a small molecule by removing chiral centers, starting with natural amino acids and so on. So there's definitely options there. But when you have, let's say, a peptide of 20 amino acids, then probably it's not eligible. Again, if you're in a gray zone, reach out to us and we're happy to give you an opinion. We get questions a lot like, hey, you have a cool program. Can you fund diagnostics or medical devices? At this time, we don't. We only fund small molecule therapeutics, and diagnostics and devices are outside of TRxA's scope. I will give you a small sneak peek. Right now, we're only funding small molecules, but we're currently evaluating a biologics geared RFP that will come out with probably later 2024. We're currently defining what that's going to look like. So what modality, what therapeutic areas, and so on. So stay tuned for that if you're working in biologics. And then we get questions like, hey, I have a really cool compound and I've shown that it works already. I know it binds to my target molecule. Can I apply for your funding? Well, of course, you can always apply, but this would be considered very early stage and would likely be less competitive than projects that have demonstrated at least a little bit of structure-activity relationship around the compound. So never say never. It all depends on the details. But if you just have a single compound, then likely you won't fare very well in the review process. And then repurposing, a lot of interest in repurposing in the academic environment and nonprofits, and also actually within the Critical Path Institute itself. We actually have a whole program that's focusing on repurposing, not with direct drug discovery support, but more like once it's on the market, can we mine data from, let's say, electronic health records to see what makes sense to use this drug for in a different indication. But there's a very complex business model, and we don't have time to go into that all of that today. But it's currently out of scope for us. It's really hard to figure out who would fund a repurposed drug discovery and development project beyond us. So that's why we're shying away from that at this point in time. So yeah, we get the question, which I answered already a little bit. If I apply for stage 1 now, can I apply for stage 2 or stage 3 later? And yes, a successful project could progress in our pipeline beyond its initial stage designation, but we would likely tailor that follow-on funding to what you need for your project. So it won't be specifically 500,000 or specifically 1 million. It's probably going to be a conversation like, hey, the next step is really to do this really intricate animal experiment. It's going to cost $175,000, or we need this API scale up done for $325,000, right? So it's going to be much more bespoke to what your project needs at that point in time, and it will happen outside of the currently established RFP timeline and process. And then where the funding goes to. So does TRxA funding all come to my lab? Well, I want to be clear, the funds are awarded to the project. So that's maybe not your lab. Maybe part of the funds, or maybe a lot of the funds, can, for example, go to CROs or maybe consultants that we need. Although we do want to highlight that costs for CROs or consultants are not eligible for indirect costs, but still, it's whatever the project needs and whoever can execute a part of the project most efficiently. And then what are allowable costs? They are really associated with the defined project tasks like lab supplies that you need, animal procurement, core services. What we don't allow are general lab expenses. And for that, an example is when I was at the institution before, we had a lab that was funded by us, buy new lab coats for everybody. That's an example where that's really not the spirit of what we're trying to support here, right? So no general lab expenses like that. We don't support travel, equipment, tuition, or IP prosecution. Since we don't take any ownership stake in IP, we think that is the obligation of the institution to protect that appropriately. Now, and again, of note, our indirect cost rate is capped at 10%. So how many projects do we fund? So we anticipate funding between 4 to 7 projects per year in the coming years. And the success rates, I'll walk you through that second bullet. So last year, which was our first year, just as a FYI, the number of attendees at this webinar is about double as last year. So maybe that gives you a bit of a sense of what we can expect with respect to the number of applications. But last year, we received 47 pre-proposals. Of those 14, so about 30%, were selected to go under CDA. Out of those 14, 10 were invited for a full proposal, so 71%, and actually 2 ended up being ineligible because of a contractual IP situation. 1 person withdrew, and only 1 really fell out because the chemical structure was not what we would like to see. And out of those 10 that submitted a full proposal, 4 were funded, so 40%. So it depends a little bit on how many projects we get into 250,000, 500,000 to 1 million, how many we can fund. Our budget is a little bit higher this coming year than last year, so maybe we'll fund more than 4. But again, it depends on of course, the quality of the applications and strategic fit. Now, what if I don't know what stage to apply for? I would say request a consultation with us, with the TRxA team, to get advice and clarity. You can reach out to our general email address, which will be on a later slide. Kyla will get back to you. When you request a consultation, we do ask that you fill out an intake form that really specifies a little bit more clearly what the question is that you'd like to ask us, so that we can prepare for answering it when we meet. And then there's the question, well, what does TRxA get out of all of this, right? So the template grant agreement is on our website. It will define that. So the terms and conditions include a revenue sharing component. So if this becomes a resounding financial success, we would like to get a percentage of that success. So we get between 5 and 10% of the income to the institution. And that depends on whether it's a $250,000 project or a $1 million project or something in between. So that gives you a sense of where we are with that revenue sharing. And this has all been benchmarked against other nonprofits terms and conditions. So we like to think that we're actually within reasonable territory here. Then sometimes people come to us like, I really have no idea where to start to find the right CRO for some of this work. And so if you require advice on this, we can help identify suitable CROs for part of the work. We have no geographical restrictions. We can work with CROs all over the globe. We do ask that management of the contract is done through your institution, but we as a TRxA team are happy to help manage that work, meet with them together with you, follow up on things if they're falling through the cracks. So we can definitely help you with that. And then people wonder if they need quotes for their proposal or pre-proposal. You do not. We can provide you some ballpark numbers for some standard drug discovery assays, like selectivity panels or PK studies. We have kind of ballpark numbers for those so that we can help you build your budget. So when it comes time to that, then feel free to reach out to us. There will be materials on the Salesforce portal that can help with that. And this is based on our knowledge of working with a bunch of different vendors. So they're kind of ballpark figures, but at least it gives you a sense of how much would it cost to get a PK study done on 10 compounds, right? So you at least have a sense of that. And you don't need to do all that work to get quotes from different CROs without knowing even if your project's going to get funded or not. And then we get quite a few questions around IP. So do I need to have IP filed before I can even apply for your program? And you don't. So we don't have a requirement that IP protection is in place, but rather we want to make sure that the chemical matter is eligible for protection. We do expect once you do start the IP protection process, and again, sometimes it's better to do that later, right? Because your patent clock starts ticking once you file. So sometimes it's even better if you haven't filed anything yet, but it depends on the situation. But we do expect IP to be filed internationally, not just in the US or North America. And that's just, again, for business reasons. When you have future licensees or investors that are looking at your program, they will want to see broad protection of your IP. That's not just in the US or North America, but also Europe and maybe some Asian countries as well. So this is one of the last slides. There's a bunch of resources. I'll leave this up for a while. Our website is cpath.org/programs/TRxA. If you go to cpath.org, you can also just navigate to it if you click on programs and then TRxA. On our website, we have a guidance document that delineates the questions that we ask in our Salesforce portal. And actually, once you create an account in Salesforce, we have a Word document that you can download so that you can work on those questions and answering them in Word without having to navigate through a portal and being afraid that it's not going to save, right? So we try to make this as a low barrier to entry as possible. We also have our user guide Salesforce document that really explains how to navigate the portal. And we have that template of our agreement. And again, we highly recommend that you talk to your institution's tech transfer office or grants and contracts office to make sure that they're in principle okay with that. Our generic email address is trexa@c-path.org. So feel free to reach out there if you want to request a consultation or you have more questions, or also feel free to reach out directly to me at meverts@c-path.org. And on LinkedIn, I'm on LinkedIn, CPath is on LinkedIn. Feel free to connect with us there, where we push out when we have, for example, an RFP. If later this year we're going to push out our biologics RFP, this will all be shared on LinkedIn and of course via our email list, but we don't have a massive email list yet. So if you registered for this webinar, then we have added you to our communication list. We don't spam you. It's really just to announce when we have new applications. But again, here's some ways to contact us. And then at the bottom, we actually published an open access article recently called Successfully Navigating the Valley of Death, the Importance of Accelerators to Support Academic Drug Discovery and Development by Mark and myself. And it's been published in Expert Opinion on Drug Discovery. And again, we paid for that to be open access. So feel free to Google for that and download that. And it really talks about how accelerators can help. So let me stop there. I would love to ask the team, Michelle, Kyla, and Mark, to come back on camera as well. And then Michelle has monitored the questions, and she can, start hitting us hard with y'all's, burning questions.
Lots of questions here. So thank you, everybody, for sharing your thoughts. So a common question in here is related to various diseases. So just to remind everybody that TRxA is disease agnostic, indication agnostic. So there are no limitations there. Okay, so we'll dig into this. We're still coming in and scrolling here. What is the structure? And Micah, maybe you can take this one. What is the structure and volume of a pre-proposal for phase 1?
Yeah, so the structure, so we're not asking for a certain page limit, right? Because we have these question and answers where we have certain character limits actually that are defined for each question. And it really talks about your target, your chemical approach, your chemical structure and your optimization approach, your the uniqueness of your program, I would say. And actually, Mark might be better poised to answer what it ends up being totally when you add up all the pages. About how much is it?
Yeah, it's about a dozen questions. Most of the questions are essentially one line. There's only maybe 3 of the questions I think that have got longer answers, but again, it's a paragraph. We really want to hone in on the vital information. So I think in total it works out to be about 2 pages if you cut and paste it all together. But yeah, we're not expecting a lot of flowery language, so it's designed to be as less of a burden to applicants as possible.
Yeah, and the feedback we received last year. Oh, go ahead, Michelle.
Mark has also developed sample proposals that are available in our guidance document. There are links to that, a sample pre-proposal and a sample full proposal that will give you some insights into what that would look like.
Yeah, and I would say last year, our feedback, we survey all our applicants to make sure that we improve where we can. The feedback was really positive. That was easy to fill out, and because the questions are really clear and targeted, it tends to make answering them a little bit easier rather than like, oh, give us your background and you have 5 pages to do it, and it becomes a little overwhelming. And so we try to be very pinpointed in what we would like you to answer.
Excellent. So this is, I'm going to toss this question to Mark. Do you consider projects for stage 1 that identified a hit that is an analog that is part of a patent of an approved drug?
Yeah, and I actually didn't type the answer because I think we got this question a couple of times. It's a good question. That no, our expectation is that the screening hit would be a known compound, and early SAR would probably be known because it'd be SAR by a catalog. You know, we'd encourage you to do that and avoid doing chemistry. But yeah, so initial hits can be known, but we'd probably like you not to be in the middle of somebody else's patent. But if it's commercial compound and you can analog away and get novel IP, that would absolutely be covered. That would absolutely be in scope.
Yep.
We have a question here asking us to repeat the deadline dates. So the pre-proposals are due on March 31st. And we will, make notifications on decisions the week of May 13th. And that's for those who would be selected to advance to the CDA phase. That would happen immediately after. And then full proposal invitations would go out, invited to submit a full proposal. Those invitations would go out the week of July 1. And the full proposals are due on August 26th. We expect to have decisions and award notifications made in mid-October, and that's when we would initiate the project agreements with the universities. Micah, can you talk a bit about how we handle indirect cost?
Yeah, so we limit indirect cost to 10%. CRO costs and consultant costs are not eligible. So when we actually have a budget template that we provide that can help you navigate through those maths. And it's something that we benchmarked against other nonprofits. So that's something it's pretty typical, and that's our requirement. So 10% max. So the total needs to be at that 250,000 or 500,000 or 1 million, including your indirects. So make sure you don't apply those indirects to any CRO cost.
Excellent. Thank you for that. So how do we reach out to a TRxA representative if we have questions about what stage we should apply for? You can reach out to us at our email address that's displayed on the screen, TRxA@c-path.org. Okay, we have an attendee here who has several projects, 3 to be exact, that would fit either stage 1 or stage 2. Can an applicant apply more than once, Micah?
Absolutely. Yep. No, we've had that same thing happen last year. I think we had one PI who was actually part of multiple proposals. Sometimes he was a PI, sometimes he was a collaborator. We're really judging by the merit of the proposal. So multiple per PI is possible, multiple applications per institution as well. And actually, last year when we decided who to fund, the PRB, the Programmatic Review Board, actually did not even look at what the PI's name was or where the project came from. Is really based on the scientific merit. So I would not worry about that. So you can apply multiples.
And we talked a bit about the TRxA team being available for consultations. Kyla, can you share with everyone the process for getting such a consultation scheduled?
Absolutely. So if you are interested in having a pre-submission consultation with us, you can reach out to us at TRxA@cpath.org, and I will send you a link to our pre-consultation form that provides a place for you to outline some questions before that meeting occurs. And then once I receive that form response, I'll go ahead and initiate the scheduling process, and we'll get you scheduled for a 30-minute consultation with the TRxA team.
Great, thank you, Kyla. And we encourage you to be as detailed in that as possible in terms of the type of information that you're seeking, because if there are times we'd want to bring in specific subject matter experts to make that meeting as impactful as possible for you. We have another question about CROs. I'll toss this one to Micah. Can CRO charges be paid directly to CROs, which for this attendee is highly desirable because it would avoid university demands for indirect cost payments for funds passing through the university?
Well, Michelle, if I would have read that question, I would have tossed it to you. So I would say we can be flexible. We do try to avoid us managing the contracts with the CROs because it becomes very burdensome for us because we have more than one project, right? What we've encountered mostly, if somebody already has an existing relationship with the CRO, it tends to work best to just keep using that if you're happy with their work, of course. And that means your university already has an agreement with them in place, and we can reimburse the institution for their costs to the CROs. Now, when there's different fees that are added onto that, then I think it's something that we need to look at together with your finance department, what we can do to reduce those fees as much as possible. And we just have to kind of look at that on an ad hoc basis. Michelle, anything you want to add there? Because this is exactly something we actually just went through with one of our grantees.
Right. In one of we actually, when it comes time to submit a budget, that would happen in the full proposal stage. We have a budget template for you to help make that as easy as possible. And indirect, we don't, it's designed so that we don't apply indirect charges to CROs. So because we feel the same way, we want the funds to go to as much research as possible.
And there's a question about just continuing the CRO about, can developers subcontract external services for pharmacological assays? And the answer is you can apply in fact, you could apply all the funds to CROs. So, anything that's not available within the university, we encourage you to engage CROs for.
And we have just a few minutes left. Rest assured, if we don't get to your question, please email us at TRxA@cpath.org, and we will answer your questions individually. Is it possible to advance from stage 1 to the next? If the project goes well, what would that look like? Micah?
Yeah, we actually just defined that actually. So I alluded to it during the talk, but we will not make you go through the RFP with everybody else, because at that point in time, we know you, we've established a relationship, and we have a sense of, is this project going anywhere, yes or no? So there's a tiered decision tree there at the end of your project. First, there will be an administrative review, like, did you submit your report? And there's some things like, are you a good team player? And if the answer is yes, which hopefully they will, then there will be a request, okay, what's that next scope of work look like? And again, this does not have to necessarily be a perfect 500,000 or 1 million. In fact, because we're going to be very collaborative, we meet each other monthly, there will be much more better sense of, hey, this is really the next scope of work that we need to do, and it ends up being 375, then we'll just get 375, right? So what we'll do is, together with you, put that next scope of work on paper that gets sent to our external scientific advisory committee, because we want to have that external set of eyes. So they will look at your progress reports as well as that plan for the next work. They will give an assessment, does it make sense or not from a scientific and technical perspective. And then we have our programmatic review board again look at, okay, based on our portfolio, on the availability of funds, can we continue supporting this project, yes or no? So it's a little bit outside of the regular RFP process. So it's going to be much more nimble, and the idea is quick. To make sure that we don't have a gap in any work between, let's say, stage 1 and stage 2 or whatever. I think at that point in time, we kind of go away from the stage names. It's really just whatever the next piece of work is that we need to do to add value to the project. Now, we would like to see things like you've engaged with external groups to know what really are the outstanding questions that would add the most value to get a highest chance to get a licensing agreement in place, for example. So there's a couple of different aspects in addition to the scientific and technical parts that we all work together as a team during that first year that we work together.
Great, and I'm going to just wrap up with one last question, because this is an important one that we've seen in the past. If a target is involved in multiple diseases, is it allowed to target multiple diseases or just select one first?
I'll take that one. So yes, I would suggest select one first. We've actually have projects come to us where they say, hey, we can use this for Alzheimer's, Down syndrome, and glioblastoma. Isn't it amazing? And I think if you're looking at hypothesis-driven research where that type of biological exploration can apply to multiple therapeutic areas, that's a good thing. When you're talking about drug discovery and development, focus is the name of the game. So I think you need to, and again, we can help wherever we have knowledge. What is the best indication to do first? And that could depend on the patient population. So how easy or hard is it to do clinical trials? What does the disease progression look like? So how long should a trial be? All of those may play into the decision what your initial indication is going to be. So then you focus on that. Now, you could address that potential to apply to multiple therapeutic areas in your write-up a little bit, but avoid going on 2, 3, maybe even more multiple parallel paths, because then you make 1 millimeter progress in 1,000 directions, rather than focusing all your energy on indication 1, knowing that once you get to IND and beyond, then you can start working on those secondary indications at that point in time.
Great. Thank you so much, Micah. And thank you everyone for taking time to learn about TRxA today and for the great questions. Again, if we didn't get to your question, I know Mark is typing furiously trying to answer everyone's questions, but please reach out to us at TRxA@cpath.org. And this webinar has been recorded, and we will make the recording available if you want to revisit the content or share it with a colleague. Any closing thoughts, Micah?
No, I'm just really excited that we have this RFP again. It's been a lot of fun last year with our first cycle. We learned a lot, so we're applying these lessons to this round. And again, happy to talk with anybody that wants to reach out and wants our opinion on things. And thank you for attending.